RNA sequencing, also known as RNA-Seq, is a technique for investigating an entire transcriptome, which is the pattern and level of expression of all genes in a particular cell or tissue. For example, it is a way to compare the gene expression differences between normal and tumor cells. In this procedure, RNA pools from the different samples are converted into double-stranded cDNA molecules. Although a human cell has about 20,000 genes, let's simplify our analysis to just 3. We want to know how many of these cDNAs correspond to each gene so that we can compare the transcriptomes of the normal and tumor cells. With RNA-Seq, we can find out that gene 2 is overexpressed in tumor cells, while gene 3 is underexpressed. After the cDNA is produced, it is sequenced using a next-generation sequencing technique. Next-generation techniques vary, but usually consist of several steps. First, the DNA is fragmented to generate smaller pieces appropriate for sequencing. The fragmentation step, alternatively, can be done earlier on the starting RNA sample rather than here at the DNA stage. The ends of the fragments are then modified by the addition of adapters. These adapters allow the DNA fragments to be amplified by PCR, which generates enough product material for sequencing reactions. Each amplified product may be sequenced from both ends or from a single end using primers and DNA polymerase. The resulting sequence data are aligned with the reference genome. The transcript copy numbers and length distributions are analyzed and compared between the two samples. Many researchers are transitioning to using this technique of RNA-Seq and away from using a similar method developed earlier called DNA microarray analysis. In brief, in DNA microarrays, the two mRNA samples are processed in a similar way. However, the cDNAs are fluorescently tagged to distinguish the two pools. The cDNAs are separated into single strands and added to a microarray, where they hybridize to complementary oligonucleotides affixed to each spot. Each oligonucleotide corresponds to a known coding region of the genome. Unhybridized cDNAs are washed away. The resulting fluorescence indicates gene expression levels. Red means increased expression of that particular gene in tumor cells compared to normal cells. Green means decreased expression in tumor cells. And yellow means high expression in both types. While DNA microarrays and RNA-Seq produce similar gene expression profiles, RNA-Seq has several advantages. The first is that unannotated genes, which are genes that have yet to be identified, will be represented in the RNA-Seq sequence data. In contrast, all of the oligonucleotides on a DNA microarray are chosen based on known gene coding regions, and so unannotated genes are not included. RNA-Seq also identifies any RNA splicing variants that may be produced during gene expression. RNA-Seq also has a wider dynamic range. That is, low-abundance transcripts that wouldn't be bright enough on a DNA microarray can be identified with RNA-Seq. At the other end, high-level transcripts that might cause signal saturation in DNA microarrays are fully counted in RNA-Seq. As RNA-Seq becomes cheaper and more familiar, it will likely eclipse DNA microarrays as the method of choice for transcriptome analysis.